Welcome, students, to week 19 office hour, when we are going to talk about gene expression, the transcription and translation. Basically, the DNA has the code, and that is going to be transcribed and translated into amino acid sequence, providing and supporting the protein's primary, secondary, tertiary, and quaternary, if it's applicable for the protein, that it can function. Now, we discussed at the beginning of the semester the importance of the protein, but let's recall it, why they are so important. They are the working horse in our cells. Where can we find proteins? Let's see. We can find them. Antibodies are made up of proteins. We can also find them in enzymes. Enzymes are made up of proteins, enzymes. So we are talking about metabolic enzymes, ATP synthase, that's all a protein. What else? The DNA polymerase, RNA polymerase, helicase, topoisomerase. These are the ligase also made up of proteins. They are also structural proteins like the keratin in the hair or the actin and myosin in the muscles. Protein can be the hemoglobin that carries oxygen all around in our body. They are transport proteins, transport molecules like the ATP the sodium potassium pump, or they are also, the ion channels across the cell membrane, or the aquaporin that transports a high amount of water from one side of the cell to another side. Also, they are regulatory proteins in cell cycles, like the cyclin or the cyclin-dependent kinase. Recall those. We already discussed them, so it should ring a bell at this point. So at this time, now get back into the flow of information. We have the DNA. And the DNA is going to be, let's see, it's going to be turned to RNA, messenger RNA. This is transcription, called the process, when the DNA language is going to be an RNA language, going to turn into. And the other one is the translation. That's a different language, the amino acid sequence language, the protein language. So this will be the protein. At this point, we stop at the protein, but next office hour, we will talk about the protein modifications. So, because that's not how it is right away, or we can also modify the proteins. Now, if I would say in the prokaryotes, this happens all in the cytoplasm. They don't have a nucleus, so it's not separated from the events from each other, the transcription, translation. As soon the transcription happens from the DNA to form a messenger RNA, the messenger RNA right away, it's going to be translated by the ribosomes into protein. For eukaryotic organisms, I will put a little bit here a pre-messenger RNA segment. This is going to be the transcription. This is the transcription, and from pre-messenger RNA to RNA will be the splicing. splicing, because messenger RNA in here contains the pre-messenger, some exons and introns. The exons are the expressed parts. They are the ones that they are going to be cut out from the pre-messenger RNA, spliced together, and they are the ones that they are going to go outside into the cytoplasm. This will be the nucleus at this point, and this is the cytoplasm. So, but the introns are the intervened segment, express segment. Now, so the introns, they will stay inside of the nucleus, and with the splicing, we will have the capping, 5 and cap, added to the protein, to the messenger RNA, and also the polyadenine tail is going to be given to the pre-messenger RNA to turn into messenger RNA, a mature messenger RNA, to be able to leave the nucleus through the nuclear pores into the cytoplasm. The translation is happening all the time at the ribosomes. By the way, the ribosomes are made up of RNAs as well as proteins. Ribosomal RNA. Whoa, that's so many different types of stuff. We have the ribosomal RNA, we have the messenger RNA, and we will talk about the transfer RNAs, that they are the ones that they contain the amino acid with amino acids to bring the appropriate amino acid based on the codons on the messenger RNA to build up the protein, the actual amino acid sequence. Now, how is DNA different from RNA? Let's go and discuss that a bit. In here, so the DNA contains double helix in cells, and the RNA is usually single strand. The DNA contains thymine, besides the other nucleotides like guanine, cytosine, and adenine, but the RNA contains uracil instead of thymine. Now, I would like to just share my screen with you at this point. Like this, how it is. So basically, here is the DNA, here's the messenger RNA is going to, the RNA polymerase is going to make the pre-messenger RNA. Here is the splicing going to happen, and the mature messenger RNA with a cap at the 5th end and the 3rd end, the poly A tail, will go through the nuclear envelope, goes outside. This is the ribosome, and the ribosome is going to read the codons on the messenger RNA, and the transport RNAs are going to combine with the codons, appropriate codons, complementary pairs of each other, and bring the amino acids into the codon place. They have the anti-codons in here that they match with the codons in complementary ways, like A goes with a U, U goes with an A, G goes with a C. And now, this is a weak hydrogen bond formation when they adjust themselves, the tRNAs, with the codons on the messenger RNA. And these are the amino acids that they combine together with covalent bonds to form a growing chain of the proteins or the amino acids to form the protein eventually. So that's what I wanted to show you guys. The next one is, stop sharing. So I would like to go over how to make how to use the codon table. So we are going to be here, and my example is we have a coding strand of DNA that is going to be I try to just make sure that I correctly just use those ones, and A, G. Now this is going to be the DNA. Now we are going to use a template strand of DNA. That's the other side of the DNA. This is the fifth and the third, and so the template will contain anti-parallel. So T goes with an A, T, T, A, and C, A, and T, A, C, and this is the fifth end. Good. And now we are just making the messenger RNA. The RNA polymerase is going to do using the template strand to transcribe the messenger RNA. At this point, we will leave out the splicing. So it will be a fifth and U, A, U, and G, U, U, and A, U, G. Now, how to use the table at this point? I will share my screen again with you guys and go here and back. Where I will find it. Okay, this is the codon table. So what we will do is checking the codons. These are the codons in here. 1 codon, 2, 3. It's always for the first letter to the last one. So we check the first letter is a U. We have an A in here. This is the second letter. So U, A, and the third letter is U. So it's going, the tryptophan. This is going to be the amino acid sequence, TYR. This is the first amino acid. The second one is GUU codon. So we have the G in here, the last or first letter. So GUU. So that's a valine. And the third one is AUG. A, first letter, U is the second, AUG, and that's methionine. It's red because it's also a start codon, but that's okay. We do not worry about that at this point. So this is how you use that. Now, we usually don't ask, but if we want to just check the transfer RNA, and this will be the anti-codon, this would be like AUA, and this will be 3/5, okay? But we usually do not ask the anti-codons, but it's every time is complementary of each other. I think that's good. What else? Okay, so before we conclude, let's talk about the viruses a bit. And we will have a virus section eventually, but let's get introduced to the viruses a bit. So the virus, like here is a bacterial cell. So it has a circular DNA because it's not eukaryotic. And we have a virus with a DNA, for example. This is the DNA. We will just go back and discuss later on the RNA virus. This is called the bacteriophage, that when a virus infects a bacteria. The DNA is the only one that goes inside of the bacteria, and we call it transduction. When the virus gets in here and pushes its DNA inside of the bacteria. That bacterial DNA, that's a viral DNA, can integrate into the DNA of the host bacterial cell. And at this point, that DNA, viral DNA, will be part of the bacterial DNA. For example, if that DNA is coding for a toxin, in this case, the DNA that integrates in here, like I will just make it red, and I will make it red that it integrates in here. This will be a toxin-producing bacteria. Its genetic makeup is going to change due to the viral DNA incorporates into it. That's a horizontal gene transfer. Gene transfer. Now, compared to the vertical gene transfer, when the parent passes on its genetic material to the offspring. In here, it's pretty much same generation, but the DNA goes from one place to another. Now, what did I want to say in here? Okay, so this really tells us also that supports the idea of common ancestors, like the DNA code is really universal, supports evolution and the common ancestors. The DNA in the virus is pretty much be able to get transcribed and translated into a protein, because that DNA is going to be transcribed to RNA, and we have a ribosome, messenger RNA, and this ribosome is going to read the code, and this bacteria is going to be able to produce the toxin. But that code came from a virus. So this is one thing what I wanted to mention to you guys. And another one is how the retrovirus works. So we have again a cell. We have the DNA. It doesn't matter it's a prokaryotic cell or eukaryotic cell. We just say this is a prokaryotic cell DNA. And we have a virus that contains an RNA segment in here. Plus, it has an enzyme, reverse transcriptase. So two things, RNA and reverse transcriptase. This virus enters into the host cell. And in the host cell, we have the protein, the reverse transcriptase. I will just use a color stuff and the RNA. So the RNA goes here and the reverse transcriptase. And now this reverse transcriptase makes this RNA transcribed reversely into DNA. And now that DNA can integrate into the host DNA, and the host DNA, as it's going to be transcribed to messenger RNA, and it's going to be translated to protein, and this protein can be a reverse transcriptase again, and the messenger RNA, some parts are not going to be translated into protein. So these are going to be viral particles. So the host cell is going to produce more and more viruses. That's it for today. Have a good one.